Hello, my name is Angie Peltier and I work for the University of Minnesota Extension out of Crookston. Today I'm going to introduce you to a project in which we are investigating whether a new way of applying foliar fungicides to manage white mold in soybean can improve fungicide coverage, efficacy, and crop yield. White mold is considered a disease of high yield potential soybeans, meaning that many of the practices that farmers have adopted to allow soybeans to better realize their genetic yield potential, like pushing planting dates earlier, reducing row spacing, and increasing seeding rate, can result in a fuller canopy and a cooler, more moist environment under that canopy at flowering, which can increase disease potential. As with any plant disease, three things must be present at the same time in order for white mold to occur: a susceptible variety, a virulent strain of the pathogen, and environmental conditions that favor the interaction of the crop and the pathogen. In our experiments, we planted susceptible soybean varieties adapted for each trial location. In the case of white mold, while some soybean varieties are more genetically resistant to disease than others, there is no complete resistance. And so when the pathogen is present and environmental conditions favorable, disease and yield loss can still occur. Spores of the fungus that causes white mold land on senescing flower petals, using them as a food source to then infect the plant at one of the lower leaf nodes. Proper timed foliar fungicides can protect a crop from some disease and yield loss. But because these fungicides act as protectants, incomplete coverage at those lower leaf axils where infection takes place means that some yield loss will still occur. To improve their crop's nitrogen use efficiency and reduce the amount of nitrogen that's lost to the environment, more and more corn farmers are choosing to side dress at least part of their crop's nitrogen needs into a standing corn crop. Companies have developed sprayer nozzle bodies that can attach to side dressing equipment, hold 3 to 4 nozzles, and ride between rows and down within the canopy. We hypothesized that a sprayer boom that allowed 3 nozzles to ride between rows and within the canopy would significantly improve fungicide coverage, efficacy, and would result in higher soybean yield in a high white mold environment. The pathogen that causes white mold produces hard, melanized survival structures called sclerotia, or sclerotium in the singular. Because this pathogen is also capable of causing disease on many of the other broadleaf crops grown in northwest Minnesota, like dry edible beans, canola, and sunflower, there is a high likelihood that the pathogen would be present in our trial locations. But to be safe, we chose to infest the trial locations and put the pathogen where we needed it to be. The first thing we needed to do was to prepare some media to multiply the pathogen. We essentially produced and poured a sterile vegetarian jello filled with fungus-friendly nutrients into Petri plates. We worked inside a laminar flow hood to reduce the chances that our cultures would become contaminated with anything besides Sclerotinia sclerotiorum, the fungus that causes white mold. Working in the hood, we transferred little squares of the pathogen from a plate where it had been growing and onto new plates to ensure that it was both still alive and to replicate it. As we gave our cultures some time to grow in their Petri plates, we made preparations to produce even more of the pathogen. Our plan was to grow it on moistened sorghum seed. Because we also wanted to make sure that we were only adding our pathogen of interest to our field plots, we used an autoclave to sterilize the sorghum seed. Autoclaves use heat and pressure to kill microbes. After about a week of growth in their Petri plates, we chopped up the contents of one Petri plate and added it to the sterile sorghum seed, sealed up the bags, and gave the pathogen time to colonize the seed. After close to 3 weeks, we broke up the colonized sorghum seed and transferred it to sterilized paper bags in order to air dry for several more weeks. After applying our fungicide treatments, we then broke up the fungus sorghum seed mixture and spread 1 liter of it onto plots in the trial. In order to create a moist, humid environment that favors white mold infection, the plots are mist irrigated hourly each night. Before applying the fungicide treatments, we placed water-sensitive paper at 6 and 12 inches above the soil line in the soybean row. Using a scanner and a computer program, we will be able to analyze the spray pattern captured on the water-sensitive paper to measure fungicide coverage. As we get closer to crop maturity, we will also collect data regarding disease incidence and severity and soybean yield and quality. This project was funded by Minnesota farmers and their checkoff funds through the Soybean Research and Promotion Council. Thanks also go to Jeff Nielsen, Ashok Chanda, Jason Brantner, Joe Waderick, and Dean Malvick for their contributions.